asked what are the advantages, disadvantages of light and electron microscopy for visualizing neurons. So it's always hard to be the first one to say something. You should not be afraid to say nonsense. It's totally normal that when you're learning stuff that you're not sure about things. And some of the questions actually don't have a clear answer, right? So there is even many questions where there's no clear right or wrong.
So can I say something?
Yeah, just go ahead, yeah.
So one advantage of light microscopy is that we can use proteins like GFP to tag certain populations of neurons or certain parts of the neurons. Well, we cannot do this in electron microscopy, but in electron microscopy we get way finer details on the structure of everything.
Yeah, that's correct. Electron microscopy kind of allows you to visualize everything, right? You're not missing anything, but you can't tag particular proteins, and so you cannot really tag particular structures. So the advantage of electron microscopy is clearly you have a higher resolution, and the disadvantage you see almost too much. It's hard to focus on any particular thing because there's so much structure that is visualized.
One question for electron microscopy. Can't you do like immunogold labeling?
Yes, okay.
I'm not sure whether you can do it with as much things, but...
Yes, it's correct. To be precise, it is possible to do immunogold labeling. In principle, you could even, use different kinds. You could use nanotags, right? And you could, in principle, also do multi-channel labeling. However, in practice, when you do electron microscopy, you have to fixate the brain, and you have to... there's this step where you put glutaraldehyde, which cross-links all the proteins, which means the immunoreactivity will be quite poor. So it will be, typically it's not very as precise, the immunoreactivity is not as precise as you can achieve in brain tissue that is fixed for light microscopy. So you get much better quality immunolabeling. Yeah. But it's in principle possible, to label structures.
You have the small smartwatches.
I think another disadvantage would be that you can't do it in vivo, so you have to.
Yes, also exactly. You have to, if you want to watch a process in a time lapse manner, right? You need to kill animals at all the points of interest, and you cannot look at one individual brain evolving in time. That's right. You can't do it in vivo. That's another disadvantage.
When people say Internet of Things, it usually means the interconnection.
Okay, very good. No more questions. If you have other questions about any of the lectures, go ahead. I just put some. It doesn't have to be that these are the most important ones. But I'm not sharing my screen anymore. Okay, I'll share it again right away. Here's another set of questions. What does this is from the Systems Neuroscience lecture, what does the neural circuit of a reflex tell us that goes beyond evidence for muscle contraction? Right, we know that a reflex, like tapping on the tendon below the kneecap, leads to the leg, the extension of the leg. And what did we learn in class that tells us that goes beyond simple muscle contraction? Do you remember that?
There's also an inhibitory interneuron that leads to a relax of the extensor muscles.
Exactly. The relaxation of the antagonistic muscle of the other side of the joint. So the brain seems to have this push-pull architecture of not having muscles. In this reflex, the muscles don't compete with each other, right? It's not that if they're both stiffened, then they will just stiffen the joint, and that's not what the reflex does. It really relaxes the opposite muscle. And this is seen from both the electrophysiology and the neural circuit. And the goal of systems neuroscience is to bring together this kind of circuit view and the functional view from imaging and electrophysiology. I had only one slide about computational neuroscience. So in this class, I will. The first few weeks will be mainly following this book. You know, the book. Following the book of Purvis, the neuroscience book. So it will be mostly about the basics in biology, and the class will get more and more mathematical. You'll see. So we'll be staying mostly with computational concepts later on. And so what do you think? Why is computational neuroscience important, and why will it be even more so important in the future? That's one of the questions. There's no clear right or wrong answer, but kind of something to think about.
I would argue that it's nice to have a rigorous mathematical computational model for mechanisms. It gives a really clear definition rather than just describing it in words.
Okay, yeah, it's nice. Yeah, it's nice to have.
And you can use it for other things. Like, for example, in neuromorphic engineering, you can train spiking networks, modeling networks.
Yeah, you can use it, you can apply it, sure, it's nice. But is there a more fundamental reason why it's important, really necessary, you know, why we cannot live without computational neuroscience?
Prediction.
So I would say we're getting more and more facts about the brain, right? We're still in the age of fact collecting. We don't have a universal theory of the brain, right? So there's many papers that are written in these high-profile journals about surprising findings one didn't expect. Right? And so we're still in an age of, so we will need more and more a kind of theory to bring these facts together, because we cannot just remember all the facts and make a list of facts, and that's our understanding of the brain. They need to come together in a very coherent, reductionistic way. So, and that's why the more facts we collect, I think the more important computational neuroscience is going to be.
They are quite big. They're the size of the centimeter.
Any other thoughts
So you cannot untest
about this?
It could revolutionize our artificial intelligence, maybe?
Yes, it's true. Right? I totally agree with that. I think AI, we're going to see artificial intelligence, the biggest breakthrough. They really come from people trying to understand the brain, right? Trying to understand the hierarchies, the deep layers in cortical processing, and trying to understand the mechanistic reasons for operant conditioning, classical conditioning. And these theories gave rise to, deep learning and reinforcement learning. And so, totally agree with that. This is the driving engine of artificial intelligence.
Also, maybe it reduces the need of animal experience.
Yes, also good, yeah. We're still not at the age where this really can be reliably done on many levels, but because the brain is meant to control behavior, and as long as we don't understand this link very clearly between the mechanisms in the brain and the behavior, we still need the animals. But it's definitely foreseeable a future where understanding of the brain is so good that we hardly will need any experiments anymore. Maybe 50 years from now, who knows? Okay, any more thoughts about this? Okay, if not, I have, I think, one more.
I can share an additional thought if I'm allowed, Richard.
What?
On why computational neuroscience?
Yeah, please go ahead.
So, just like if you describe a computer based on its kind of electric properties, you're not really describing a computer. And in the same way, the brain won't be completely explained if we just describe all the physics, even if it's with a few simple laws. So the computational route allows you to give a more complete interpretation of the functioning of the brain as a kind of information processing unit rather than just a physical blob of matter.
Right, yeah, it's definitely the currency for explaining the functioning of the system, right? You would say without it, we don't really have an understanding. Yeah. Then what question I had, that's on the action potentials. Yeah, what simple nonlinearity describes the input-output relationship of a neuron? Like what, in the most simple terms, if you had to reach the operation of a neuron to a very simple concept, what is it?
A threshold function, integration with a transfer function, with the transfer function.
Integration with a transfer function?
No, threshold.
Threshold. What does the threshold do?
Well, if we are above a certain level, we get 1 as a response, for example, and if you are below 0.
Yes. There's definitely this below 0. If the input to a neuron is not sufficient, then the output remains 0. And then beyond, above 0, it's, you could say the binary neuron is the simplest one. Or you could say a linear neuron is simple, or you could say a thresholding, a saturating function is simple. This will depend. So we will derive, the equations of a rectified linear unit. I guess some of you have heard of rectified linear units, ReLU. It's very popular in AI these days. And so we will derive the ReLU equation from the biophysical properties of neurons. That will be in a few weeks from now. Yeah, and then, so why do neurons exhibit action potentials? Clearly, there's no right or wrong question, because evolution just decided to do it. Nobody knows the reasons for evolution, but it can still speculate about this question.
Do you mean why spikes and not why, for example, rate or analog signals? Or because I would say so two things. So the first one is just information processing and, regulating global mechanisms. But then we also learned that it can also be used for regulating intracellular things, for example, like calcium concentration.
Yeah, so you're saying it's useful. You say spikes. The question.
It's useful for global tasks, but it's also useful for the cell that fires the action potential itself.
So there are several.
Yeah. I mean, I think there's a better reason why it's really necessary. Where you can't live.
I think to communicate over long distances, we need a sort of digital signal that can be refreshed instead of like decaying analog signals. And also I think the internal processes, which was meant before, is like more of a homeostatic perspective that the cell maintains its certain maintenance frequency, firing frequency, which allows it to undergo certain homeostasis.
Yeah, so I definitely agree with the first part of your statement, right? Nature would never have invented giraffes if there weren't action potentials, right? Because how would the brain be able to compute with the leg 5 meters away, right? With the passive conduction within an axon, with the electrotonic lengths of maybe a few millimeters, we're going to see the equations for the cable equations and the electrotonic length. It would not be possible to communicate with the muscles that control the legs. So a simple proof, you could say, is that there are some animals that do not exhibit action potentials, like the nematode worm C. elegans, right? It's a tiny little worm or so, a millimeter or so, and it has about 300 neurons and no action potentials. So it is not really necessary in small living organisms. You need it really for the larger animals. And then clearly it has probably some additional functions that's useful, that for processing, for preservation of information and so on. But I would say the main use why it was invented by nature is probably to make larger things, to control of larger things. Okay, anything else about this? Maybe if not, I'm not sure whether you have been really starting the lecture at 9 and you want a break or no. If you want no break, I would suggest we would go over to the projects and have the TAs present the projects. Does anybody have an objection? Does anybody need a short break right now? I don't know whether you started watching at 9 and you're in need of a break.
Finally, wake up
nobody has a wish for a break, then my proposal would be is that the TAs, they present the projects, and then you start thinking about teaming up with some colleagues of yours, groups of 5, and then you will work on the project. So the idea of the projects is the common idea is that so they should allow you to just do some hands-on research on some very concrete topic, and they should allow you to learn to program a bit in MATLAB and Python. Some of you might be experts already, but I know many of you will not be very experienced in programming, so we'll have a very wide diversity of students usually, because some of you are electrical engineers and others are biologists, and so we are trying to make it convenient for everyone to be joining this course. And so the projects have 2 parts. Part A, that is more strictly defined. In part A, you will just solve a concrete problem to get you warmed up, to get you used to thinking about the topic. And in the part B, you'll be more free to come up with your own ideas and to solve a problem using your methods. And we'll have a presentation in the last week of the semester. That's usually in the end of May or so. You'll present the outcome of your project. And I would suggest we even have a short presentation earlier on in the semester. We will think about that probably in sometime in April or so. We'll make you present just what the aims are that you're trying to achieve, and then you have 1 more month to just finalize the aims and present the project. And then afterwards, you have maybe 1 more month or so till end of June to hand in your report. And the report will have to be such that we can clearly extract a grade for each person for the project. So there will be a common grade. We'll try to grade the project as what is the common the achievement, and then we'll just up or down grade the individual grades by rounding up or rounding down for the individual participants, depending on their contribution. The project is graded and contributes, I think, by 1/4 to the final lecture grade. And the lecture exam will be a written exam, 2 hours, where you will have to answer questions that are not very heavy on mathematics, but that are just asking the general understanding.
Is the exam in August, or is it at the end of the semester?
I think it's in August. It has always been in August, yeah.
Okay.
We will distribute a practice question in the exam, so you can see what it's like. And I'll say more about the exam later on. But the exam is a bit like it's an oral exam turned written, where the questions are a bit like you would ask in an oral exam, and you should be able to just answer in 1-2 sentences. And for those of you who are biologists, I think they have to catch up a bit on programming to get into programming. Maybe you need to know about algebra and analysis. We'll do some, we'll see differential equations, we'll see Fourier transforms, we'll see eigenvalues, we'll see things like that. So you have to get refresh up. We see probabilities, right? We see expectations. I'll do some introductory courses for some of the math we're using, but many of the things I guess you have to catch up. Okay, so we have the projects now. Let's start with the project presentation. We have one project on analyzing behavior. Right, behavior is very complex. We know behavior is very nonlinear, and it's difficult. It's a challenge to analyze behavior. We have a project about the development of the brain, so from gene regulatory networks. We will have a project that will try to characterize the development of the brain. Then there is a project where we have recordings from neurons in the brain, and we'll try to understand how these neural recordings, they relate to behavior. It's one of the general, topics in systems and computational neuroscience to understand this link between brain and behavior. And I think we have three topics. Or do we have four? Sorry, I just forgot. Let's see if we check the
working on the exercise project. You will form a group of 3 or 4 people to design one of the state-of-the-art designs in one of the field. And while you're doing so,
oh yeah, we have a first one. Sorry about that. Yin Chang. I couldn't see his face, that's why I didn't think of it. We have a fourth project that deals with the relationship between birdsong and language, right? So it is commonly believed that language is a purely human thing, right? But it's believed that there's ancestors for language in animals, and so we're going to be testing particular hypothesis of the similarities, the precursor similarities of language found in animals. Okay, let's start with the project. Who wants to go first and present the project first? Let's see. Is Yin Chang here?
I can go first, no problem.
Yeah.
Can you guys hear me?
Oh, now I see you. Okay, hi Yin Chang.
Hi.
Okay, good. Please go ahead. You can share your screen.
Sure.
That's why I didn't see you, because of the mask, I guess.
Okay, can you guys see the slides?
Yeah.
All right. Hi everyone. I will be one of the tutors for this learning system lecture this semester. I'm Yin Qiao. I'm first year PhD student supervised by Professor Carlos. So the topic I'm offering you guys on the project, it's a sequence generation with the advanced sampling strategies, especially link behaviors between the recordings and natural language processing. Right. So in this topic, you will be starting with the sequence sampling strategies and analysis approaches, and then finally in the part B of this topic, you will apply them to generate text in natural languages, right? And what you need for doing a project in this topic is that you're gonna have like some basic understandings in probability theory, as well as in machine learning, and of course, because we're doing programming, you're gonna have some basic programming knowledge in Python to be very exact, metrics, operations, and data structure. But if you're a biologist and you have no ideas of what Python is, no problem. We will provide you some simple tutorials to guide you into the program, right? And what are you going to learn in this topic? You will gain your first experience in sequence analysis, natural language processing, of course, as generation. And you will understand different sampling strategies and implement your own advanced version. You will also capture the philosophy of generative process in various accounts. You will understand how the sequence is generated and how a natural sentence is produced by setting up a machine learning model and pipeline. The next one will be you will bridge the gap between the mathematical theory and actual implementation. We will provide you a very smooth how should I put this? We'll give you the opportunity to link your knowledge in math and actual implementation to create something that is really useful. Now, as Richard said, we have like basically 2 big parts for this topic. First, you're gonna do an exercise, and then after an exercise, you will be engaged with a actual with the actual project that leads to something. In the exercise, we have basically 3 parts. The part A and part B will be related to sequence generation. In this case, you need to understand and implement some basic machine learning models to generate sequence and analyze them. And in the second part, you have to visualize the findings by modeling one of the metrics of vector information decay as a function of binary distance between the states in sequence. As for the second part, this is where your idea shines. You can apply this approaches you learned in the exercise to generate lecture language text, right? And you can also try out different sampling strategies like sampling in reverse composition, or you can just switch between different modes, etc. And you can also integrate parameter-driven or decision-driven learning into the generation process. Finally, you can compare your approach to the AI-driven language models and then discuss the difference. So for this part, don't hesitate to be creative, because this is where you bring your own ideas to the project, and we will find a solution to the questions you're going to have together, right? So I guess this is basically the main content of my part. If you're passionate about machine learning and artificial intelligence, and you want to decipher the things, the language and sequence, then I guess this might be the correct project for you. So it's time to sign up, and feel free to write me an email at any time. Thank you, guys.
Can I just ask what the links are with neuroscience for this project?
Okay, that's a very, very good question. You guys are going to learn about a lot of computational stuff in the neuroscience, right? And actually, we're doing these things because we will start with the exercise, which will be based on actual neuroscience data set, where we will hands on the recordings of some birds and human speeches, where they're just they're collected in a special way, and then you have to analyze this data from there, and then transfer the learning into some actual experience in the natural language. So, this.
If I can already, I can add to that. Remember, only humans have language, right? And remember, we cannot easily stick electrodes into human brains. It is done in epileptic patients, and so we know a little bit about what neurons do in the human brain, but I think we will always have big gaps, especially for language. You know, when a surgeon in a epileptic patient needs to remove some brains, right, a structure, and he will always try to avoid the language areas. They are sacred, right? This is one of the most important centers in the brain that the surgeons will at all prices try to avoid, which is that we will probably not know much about really the neural code of language in humans ever. It's just it's like an impossible topic. But still, you know, we know a lot about language. Language has, just by looking at the structure of language, right? Language has this Markovian contribution. That's the green network shown here on Yingqiang's slide, and it has a hierarchical structure. So, right, and it's an interesting. Language is a mix of this Markovian and hierarchical structure, and how are these mixed, right? And how could a network of neurons achieve that? This is a topic of cognitive neuroscience, right? So it is definitely a neuroscience question, because we want to understand this the structure, you know, of language, both in terms of a neural mechanism, and so we have to because we cannot measure the neural mechanisms in humans, we have to make best guesses.
I see. And if I say something to this question, is that we should really consider the generation process of language. It's like when you're speaking, you're using your brain to fetch those words from your memory, and then construct them into individual sentences, right? And then in the computational area, then the sentences are generated, I don't know, perhaps in a similar or distant way, right? Like, you have to understand how the generation process works by how do you generate a sequence to form an individual sequence. And I think this process will, of course, it will link to the actual process where it happens in the brain. But I think this will be a really good start to understand the process.
Any more questions? If there are no further questions, maybe Yinchang, you can unshare your screen and somebody can go next.
I can go?
Thanks a lot.
Let's see. Do you see the screen?
Yeah.
Can you hear me?
Yes.
Okay, so I'm a PhD student also at the Institute for Neuroinformatics, working with Rodney Douglas and Richard. And the kind of central paradigm behind what we do is, or what I do and we do, is to try and explain the kind of immense complexity of the brain by looking at how it came to be from kind of humble beginnings, simple beginnings. As was mentioned in your first lecture, it starts with a single cell and this immensely complex kind of proceeding to a whole brain, so it's a developmental process. And in this project, we'll be looking at one specific aspect of that.
For the design project,
is there the technology that you have defined? So the data we'll be working with looks kind of like this. So this is a mouse brain, and you can see it's composed of all these individual squares. Here you can see it. And these squares are measured voxels, so 3D pixels of gene expression. And this was measured by the Allen Brain Institute. So each of these voxels contains about 2,000 values of genes that are expressed at that particular location. Now we can see gene expression kind of as the state variables of development. They're subject to a very strict control regime by the gene regulatory network. And so by looking at gene expression, we can know a lot about, or we should be able to predict a lot about the phenotype of the cells in that region. Now, this is the brain at P28, so this means 28 days after the mouse is born. But the Allen Brain Institute provides also data for all these other time points. So, for example, this is the brain at P14, so 14 days after it's born. And this is E18, so this means 18 days after fertilization. This is really an embryonic brain. And as you can see, as we step through these times, the brain kind of changes shape. Now, the shape of the brain is not just a kind of after effect, but it really has some kind of functional significance, probably. So the shape of the brain, like if the brain was shaped as a very, very long tube, it would be impossible to create all the connections that are necessary. So the shape really has some kind of significance. And what we'll try to do in this project is to build a simulator that is going to, from this shape, take the gene expression as input information, predict the next shape, compare whether the prediction was accurate or not, again look at the gene expression, try to predict the next time point, and so on. So really try to predict the progression of shape based on gene expression. Now, so what does that mean practically in this project? Well, first we're going to just look at the gene expression data. This is kind of the part A. We're going to look at gene expression data, how to pre-process it, how to kind of think of it in terms of gene regulation and these kinds of things. We're going to be building a simulator. Now, of course, this is not a computer science course, so I will be providing scaffolds and sample code and all these kinds of things to let you effectively build your simulator. And we also have to look at exactly what paradigm of simulator we'll use. And then the third, most creative part, which is going to be the part B, the creative part, is going to be how are we going to map now the gene expression information that we have to parameters like growth rate or cell-cell adhesion, so that we can predict this change in shape. So here, depending on your expertise, if your expertise is more mathematical, statistical, machine learning-like, then we might apply such methods. If your expertise is more biological, we might look at the identities of specific genes and try to work from there. So, yeah, I think that's it for me. Any questions? Really excited about this and looking forward.
So I have a question. So there is already a simulator for this. Is that right? Or?
Well, there are simulators for developmental growth, yeah. But they all, I mean, which specific, did you have a specific one in mind?
No. I was wondering if it's feasible at all. So I was wondering if there is some simulator already there.
If there's there are simulators already. So some simulators are, just solving kind of partial differential equations. So they would, we could use like the fusion equation like things to develop this growth. We could also use models that are more kind of grid-based, think like Game of Life, if you're familiar with that. There are also simulators, one of them built built at the Institute of Neuroinformatics called Cortex 3D, which kind of really looks at physical interactions. Probably what we would do is try to keep things as simple as possible. So either use this grid-based thing because it simplifies a lot of the code to just work in a grid and not have to worry about kind of physical forces in that way, or we use some simple kind of descriptive equations like diffusion equations and just simulate the differential equations.
And these simulators also work on gene expression, right? So they take gene expression and then maybe also the spatial coordinates.
So what we'll be simulating is not, well, unless you really want to, the idea was to not really simulate the change in gene expression too much. So to really take the gene expression as a prior and then just use that to derive the kind of tangents of how the geometry of the brain is going to change. So yes, existing simulators could take into account gene expression. It depends just on how you configure it all. And the simulator that we can build will, can take into account dynamics of gene expression, but that's a whole kind of level of complexity that you must be willing to go into.
I mean, Stan will hand out his simulator. He wrote a simulator, and you'll have to just at the beginning fill in some lines of code to get familiarized.
Right.
So you'll
of course.
You'll be taken, you'll be guided. You don't have to start from scratch. And the idea is very simple, that there is, correlations of gene expression in different voxels, and this correlation is related to the growth in this particular direction. And that's the idea you're testing.
Right. So
the idea is to kind of find pretty constant relationships between changes in gene expression and changes in these growth parameters, so that we know what predicts what. Like, is that the purpose?
Well, yeah. So we don't really have dynamic data. So we don't know between P4 and P14 what the kind of temporal evolution is of all these gene expressions. So the best we can do at this point is to kind of just find that constant relation, like you say, at, for example, P14, see how that constant relation kind of expounds, and then see if that matches with P28, and so on.
And maybe one other question is that, so this is the only, so you have just one mouse embryo, right?
Well, the data is collected from many mouse embryos. So the number of genes that is measured through in situ hybridization. So it's a method that isn't very high throughput. So they had to collect data from many mice to make this atlas. So it's not just one mouse. This is registered data. So this is a kind of hypothetical mouse brain. Well, it's an actually measured mouse brain, but I had to kind of average out multiple brains to make this one geometry and one data set of 2,000 genes. Is that what you're asking for?
Relation mosaic mouse brain.
Yeah. There's overlap, of course, between the measurements, so they did do these kind of sanity checks, but it's really just like Google Maps is made out of many images that are stitched together. This mouse is kind of stitched together like that.
Okay, so you're just working with the one averaged kind of sample all the time.
So the average is over the different mice brains.
Yeah.
Not an average over genes, right?
Yeah, I know. Okay.
Yeah. So we're working with that one atlas. It's really supposed to be this kind of canonical atlas of the mouse brain. Yeah, exactly.
Okay.
Any other questions?
No more questions. Thanks a lot, Stan, for this nice presentation.
Yeah, thank you.
Then we have two more projects. Do we have a presenter?
I can.
Yeah.
Okay, so I'm one of the TAs in this project, and I'm a PhD in Richard's group, and we study songbirds. And here you see
also,
the
please feel
spectrogram
free to send me
of
some about email. Send me some emails about your information or any questions
and
related
the
to
y-axis is the
project.
different frequencies. And so you see that the bird song is made up of syllables that are repetitive. And so one challenge in working with such behavioral data is to detect and
we
to
have
chant
the first lecture
this
on the
signal
trio, which is the sensory interface.
the different
This is
parts.
actually third topic in the previous year,
So
but I have removed this one as the first topic because I think
that's
this is
the goal
more
of this
related to
project,
NLO IC design
to find
and help you
segmentation
understand things better
algorithm
at the beginning of
and
the project.
potentially also in
So,
the last part
can you
clustering algorithm that that can detect these syllables. So
in the study
we
of
start with a very
this
short
course, there are
script
three reference books that you can take a look into.
on a very simple
First one
task
is the analog
to
CMOS
get
integrated
you
circuit, which you have
familiar
used for your
with the
analog
most
IC course.
important
There are
concept,
really the sections that the
then we
AIC
move
course
on
hasn't covered
to
in the last
actually
semester, which is
a
going to be
task
also
that you might
covered through this lecture.
you might wonder
And then there's a book
what
called
it has to
Extreme
do
Low Power
with this,
Maximal
but
IC Design,
I
which
will
explains a
explain
lot of
it. So
benefits and
the idea
disadvantages
in
of
this
the device to
part
work
A
with the low
is
power region,
to
and this
predict
is going to be adopted in design process
an instance of the spectrum.
And there is
In the
finally a book
spectrogram
of CMOS design layouts and
from the
solutions.
temporal coordinates.
Unlike from two the other
The
other
input
books here,
are these
which are
windows
really a
that
storyteller
might be
in each
separated by a
subtopics
gap
of the analog circuit
from
design.
the column that we
This
want to
one, the
predict.
CMOS
And,
focus more of a dictionary.
And so you will
This one
learn
includes a lot of
linear
building blocks
regression
of
model
analog circuit. It
and
explains
that,
many of
that
them
can predict
as
this
a
target
subsection
column
that you can
by
refer
this
to
temporal
when you are
coordinates.
curious about
And we look at two variants.
it's very safe.
One is
It's not something
where
you're
we
going to
predict
do
from both has to do with
signal
this
itself
goal of
from
the
environment
project.
to a electrical voltage.
We want to, in part B, where you will work together, we want to retrain a neural network on this task, because we have only limited expert level data of Birdsong. So we use what is called transfer learning.
The issue with it
We
is that
learn
they
first a
are
similar
having a
task,
magnitude
which
order
is
of
this
microns.
task,
So
and then
when
we
the
move
temperature
to
changes
the
by 1
actual
degree, you
task.
may have a temperature sensor output
So
voltage
that's
changes by
part B. We will
few
try
microns
out
or
simple architecture
similarly to the other
of,
sensors from
of
the other
a
sensors as well.
neural
The
model
key issue here is how
and
to make
train
a
it first
ultra
on this
accurate amplifier
task
that
and then
magnifies those signal accurately so we
supervise
can
it,
digitize it using
predict
the analog to digital converter.
the
So
segments
such a sensor
in
interface is
Birdsong.
built upon a series of
And then in the
analog
last part,
First we have
you
a
are
sensor
more
that
free
converts information to electrical signal.
try out
Then
different
we have a
architectures
low noise amplifier.
and also
Usually
address
this
this
is a most
problem
power hungry building
of
block that
clustering.
amplifies
So
every
really
signal through to a
finding
video
out
voltage level,
how many
and
types
then
of
followed by a variable gain.
different syllables are in the
This one
data.
tunes the circuit element
And
inside
also
of this amplifier
characterize
so
how
you can
much
have
training
a
data is needed
appropriate swing in
or
the
how
output
well,
of the amplifier
how the
before
performance
digitizing it
changes
using
with the
analog to
amount
digital
of
converter.
training data.
And sometimes we have the filter,
And
either a high pass filter,
yeah,
high
I
pass
will
filter,
supervise
low pass
this
filter.
with
So we
two
can
other
eliminate
students,
unnecessarily
Mia
a
and
portion of
Matilda.
the frequency band
And,
that was acquired from the amplifiers,
So,
but you can
yeah,
only get
do
to
you
see the
have
narrow
any
frequency band where
questions?
you are most interested.
Yeah, I have a small question. Generally about all these projects, whether are those previous projects that have already been completed, or is it something that you're currently working on?
So this one is actually a problem that we work on. So it would be really cool if
where that neural
if you
action
also come
potential
up
spike
with
is
something,
recycling.
then you
So
know
we
that
want to
you
eliminate
can
any other
contribute
long
to
current drift
something that is actually
while you are
at the
acquiring.
frontier of what we also do
Finally,
in
before
research.
ADC, you have a driver circuit. ADC sometimes have either very large capacitance or small
I have a question.
resistance to drive
You can hear me?
the circuits associated with the ADC operation. But you need to have a strong driver to
Sorry, my
provide
mic's not
voltage
great.
filtered by
What
the,
type of
filter
architectures
or
are you
amplifier
starting to look at?
amplifier,
Or
before providing
what
them
type of
to
architectures are good at this type of
finally
one-dimensional clustering? I think.
We thought that in part B, maybe it's easier
If I
to
look,
just
we try to
use
draw
it for
a behavioral block diagram of the
just
front end.
retrain
This
the
can be
last
expressed as a
layers
follows. For instance, you have
of
an analog
this
noise
feed-forward
amplifier
network
followed by the
on
VGA.
on
Those
the
are
later
gain spaces,
data.
as we have learned in the AIC course.
But of course,
You can
the
have
idea
the
would be that
and
in
finally
part
have the
C,
VGA.
you can try out
And then,
different
that
architectures.
introduces
We can pinpoint
no
you
analog
some
circuit is noise free,
things
unlike
that
the digital
we
circuit.
would
Digital
consider,
circuit actually
but
reproduce
the
either
idea
0
is
or 1
really that you,
unless you
if
cross
you
this
have an idea,
threshold of the
then
logical
you can
boundary. You can always
can
have
explore
an infinite
your own
accuracy
ideas.
of maintaining a certain data. Okay? But here in the analog circuit, all the amplifiers,
Thanks, Thomas, for the great
data converters,
presentation. I want to add, yeah, all the projects are really current, ongoing research where we have usually done the beginnings, but not all of it has been done. So it can potentially, contribute to a scientific publication. More questions about the project on transfer learning of Birdsong?
You can put this
the idea is really, like in its state-of-art artificial intelligence approaches, people find when you just predict the next word in text, always predict the next word in text, a very simple language model, that these are very powerful engines as a pre-training, that from those neural networks that are trained like this, you can easily learn other tasks, to solve other tasks with very little training data, sometimes almost single-shot learning.
Sorry, I have one last question about this project. What's the data that you use for pre-training, and how much of that data do you have?
Yeah, so that's a good question. So we have several words that we clustered, so we would use these birds and we divide them in training and testing data. So we have like a base of recordings of bird song.
We have much more than 100 terabytes.
Okay.
The data is not the limitation.
What?
You don't need to worry that you have to load all the 100 terabytes on the machine. So your computational power is limited. So yeah, this shouldn't scare you away.
Just a pretty simple. Oops, sorry. Go ahead.
Yeah, you have that huge database, but as you have described, the project seems supervised learning.
Yeah.
Do you have all that data?
Then
labeled?
Yeah. No, we don't have 100 terabytes manually data. We have a much smaller data set of manually. We will give that to you, of course. I mean, we'll get 10% of the data, and then whoever gets the best performance, I guess, will get some prize or something.
Sounds so good. No, I was just wondering, you have like 100 terabytes of manually labeled data. That would be crazy.
No. The idea is we have hundreds of terabytes of unlabeled data, right? That's clean data. Get very.
Okay, thank you very much.
Perhaps a very simple question. Why use these deep learning techniques in this specific context? I think you run into a quick overfitting problem. Obviously, you have very good reason to use, so that's why I'm asking. Is when you have these simple, at least two-dimensional data, it's a frequency over time, right? Why use deep learning for that? Deep learning, I think, is very good for high-dimensional data, but it can overfit very quickly on low-dimensional data, such as a frequency over time bar. And I'm sure there's a very good reason.
But the data is not that low-dimensional, right? If one spectrogram column has 128 samples, and you consider maybe 50 spectrogram columns, you are of 10,000, right? And 10,000 is how people started, with the what is it called? The post office, the data set on the
MNIST?
MNIST, right? 60 times 60, right? I mean, we're definitely in the ballpark here, and some of these best models, GPT-2, right? I'm not talking of GPT-3, has an input limitation of 512 tokens, right? Would you say that's very high dimensional, 512 tokens? Well, maybe not, right? So I think we're definitely in the same ballpark as the other models.
Yes, I'm not familiar with the data, I think. So, yeah. All right. Thank you.
All the transistors has intrinsic random nodes that creates a deviation from the ideal value. This is either
Okay, there are no more questions. We have one more project. I didn't see.
Yeah, I'm here.
You're here.
Hi, hi everyone. I'm Corinna. Yeah, I'm also a PhD student in Richard's lab, and I'm working with songbirds. Just going to. Do you see this? Okay, yeah. So I got very intrigued by neuroscience when I did a course in my undergrads where we would record and stimulate neurons of a very simple insect model. And ultimately, the question I took from there is how does this actually relate to each other? That neural activity that there is in the brain and the behavior that we observe. And in this project, we are going to take a closer look at one of the, I guess, assumptions that are usually made about this relationship. And that is that there is actually, or the question whether there is continuity actually between what happens in the brain and what happens in the output in the behavior. And if you remember, the concept of continuity in mathematics basically is there's a small change in one domain, there should also be a small change in the other domain. And thinking about this more closely, you will see that for neuroscience, there are a lot of conceptual questions we have to answer in order to get closer to the answer of the question. For instance, this small change, what does it actually mean? What is the distance between two neural signals or two behavioral signals? So if you think about the neural activity in the brain, you can assume there are like 3 neurons and they have an activity pattern and the behavior is repeated, so you have many repetitions of this, and you would have to define first what is the metric between in this domain. That's, for instance, one questions to ask. And here is an example of the data that we will work with. So I recorded neural signals of the songbird in a premotor region, and we have many units now in this premotor region, and the concurrent behavior, which is the song, as Thomas said before. And for the project, we have these 2 parts where first, as I said, you will work on the conceptual questions and ask what is an optimal framework to study continuity. Like, for instance, what is an appropriate metric and how can we actually test statistically whether continuity preserves. And in the second part, which is more free, the idea is to leverage neural modeling approaches to understand better the mapping between neural activity and behavior, and to, for instance, infer latent variables, and then to test if continuity actually preserves in the context of these models. This also requires some understanding of these models and ultimately also to evaluate if a model is good or not. And in this part, you will be more free, and you will also have the opportunity to read and explore what approaches there are. We have some ideas, but we would also like to foster you to think critically about it and come up with your own ideas. Yes, that's it in a nutshell.
The way the other values are affecting the value of this moment is different from...
So I have one question. So suppose that someone comes up with a new metric, and how would you be able to validate that metric?
The relationship between the neighboring values to the...
Yeah, I think that's part of the science, I would say, that we don't know the answer, and we just have to evaluate what a good metric is, what makes sense to us, and that's based on the assumptions that we make. It could very well be that what we think are similar to repetitions of neural activity are actually quite different. So I think the objective is also in a way to to make assumptions, to explain why something is a good metric and why not, and to live with the fact that you don't know the answer, the truth really, but you have to discuss it. Does it make sense?
Yeah, I would like to add yet that a good metric is the one that gives you high continuity, right? Because the idea of this project is that if I have a neural code, right, in the brain that controls my hand, right, if I take some brain activity and it generates this movement, it would not be very good if I take the same brain activity and I add one spike, and then instead of getting this movement, I would get that movement, right? If the neural code is so discontinuous that adding one spike makes the output completely different, then the brain could not be wired up to function properly. So I would say the metric that we are looking for is the one that gives us the highest continuity, which means that by looking at the two most similar motor patterns, I should get similar neural activity patterns, where the similarity is given by this metric.
One possible problem with that is that the variables in the behavioral space, let's say, are not well defined, right? So you cannot say, like, this specific hand movement example works, but for example, you don't know in the maybe song space which tones are near together or are close together.
You can make, guesses. What are in the hand space? It's not clear. I find in the end, the behavioral metric for the motor system is the muscle command, but for the auditory system, it's the activation of the cochlear receptors, right, and basilar membrane. So we know it's the output coding, we know the input coding, and we can make best guesses. But, to some degree, you're right. We don't know whether a tone that differs by another tone by 1 octave, is that very similar? Is that more similar than a tone that differs by another tone by 3 semitones, right? Is 12 semitones difference more similar than 3 semitones? It's true, it can be debated, but we can take either assumption, right? We can find reasons why one should be closer, why the other one should be closer, and we can explore. And the data will tell us, right? The data should tell us what, in the end, what we should get out is we should get this mapping out between behavior and neural code, and we should get the similarity metric out, what is self-similar and what not.
I can ask one other question. So here, the behavior is just the voice of the bird, right?
Yeah.
Again, this is thanks.
Okay, thanks, Corina, for the nice presentation. You can, of course, maybe you can unshare your screen if you want, yeah? You can write to the TAs. I suggest TAs put their email addresses in the chat, or you can contact the students also, the TAs. You're signed up as teachers on Moodle. So you can ask more questions. You can find your colleagues, your fellow students with whom you want to team up. And then we will upload, if it hasn't happened yet, we'll upload a sign-up sheet, an Excel spreadsheet or so with all the names, and then you can sign up for the project. And then you'll just be working with those TAs, and you'll meet on a regular basis every Monday at, I think, 11 o'clock. Can you upload your presentation to the Moodle, please? Yeah, that's a good idea. That's a good idea. Thanks for the suggestion. If everyone could upload their presentations on the Moodle. I made folders. I made only one for Stan as an example, but let's see on Moodle. Let's go. I don't know, Stan, have you filled it already?
No, not yet, but I'll do that now.
Yeah. So for the others, please make a folder on Moodle and upload all your stuff in there. Yeah, there's only one, Shaping the Brain. I made one folder. To create a folder, you just go... you have to learn how to use Moodle. It took me a while, but you have to add material and then choose folder. And then add your For the more general questions about all the projects, you ask me, but for the individual topics, you ask the TAs. WhatsApp chat for the students of this course. Please join so it is easy to organize the project. But, so what is that suggestion exactly? You want for each project a WhatsApp group or what?
No, we just created a WhatsApp group for each course. So like for the moment that's general for the whole subject.
So you want the WhatsApp? You want the WhatsApp group for this course, for this class?
Yeah, I mean we do it for every class, so we don't spam the central group.
And the students do that, or who does that? I should do that?
Yes, the students. It's like for us to coordinate and ask
okay.
And ask for the Zoom link. There's always someone asking.
Fully independent random process.
For the
What it means here is why.
So yeah, what should I do? I'm not sure.
Oh no, that's for the other students.
Okay, good. Okay.
Just for the organization.
Okay, good.
Yeah.
Do you know approximately at what time or like on what day you would upload the Excel for us to fill in, like each day or tomorrow?
We had a nice Excel last year. Who made it? Thomas, did you make one Excel last year?
Yeah, I can upload it.
Can you do it again? When? She asks when?
Today or tomorrow.
Okay, great. Thanks.
So we'd get an email, or should we just keep checking Moodle?
I can send the email.
At a certain AC frequency.
I just have a question for the groups. Do we have to form the groups before we sign into specific projects?
You can, yeah. You can say, I know my group, I want to be with those people. If you don't, then we can make, we could also then see who else signs up for the same project and make the groups in a more dynamic way like that. So either way is possible.
And maybe I missed this, but how many people should there be? Will there be multiple groups per topic? Because otherwise you would be like 16 people per group.
Yes, we will have. How many people do we have? Oh, 70. Okay. Yeah, we'll have several groups per topic. That's why I counted on having the most popular project to be the one on vocal clustering. That's where we have 3 TAs. The other projects depend on the amount of the capacity of Stan and Yinchang and Corina, how many student groups they can deal with. So I also had a student project last year. I had 3 groups, and there was quite 3 groups is quite involved.
Stan, can I ask you, how many people would you be willing to take, or how many groups?
So this is the first time I'm doing this course, so I have to see probably around 3.
Actually, let's not discuss this here. I would like to have a little control of what's going on.
Okay, sorry.
I'm gonna just sign up what you want, and then maybe we'll have to, change your group membership a bit. So, but we'll try to respect your wishes, and we'll try to do it in a first come first serve way. So.
Still can have a same Gaussian distribution as if the one showing the.
Okay. Any more questions? If not, I guess I would see you online again next week at 10:30.
Maybe question about the course. Will there be any exercises about the theory at any time, or do we just have these projects?
Just these projects. Would you? So we had some exercises in the past, but in the exam there will be not exercises, and so this is not really an exercise class. It would be too much work for you if you also had to do exercises now. But I hope that's fine for you.
Yeah, well, I like when there's some questions just so that we can check we understood. So the quizzes at the end of the presentation are good. So if that could carry on or even have a few more questions, it would be nice.
Okay. I will try to. I already recorded the next week's lectures and part of the week after that, but then I'll try to bring in some questions, test the understanding.
Thank you.
I have a question to the students about this WhatsApp group. Would you appreciate if the TAs joined it too, or would you value your kind of privacy talking about the course more? So what is your general feeling on this? Or some opinion?
I don't care, you can join. Doesn't matter for me. Yeah, I think you can join.
Okay, thank you.
I have a question about the meetings at 10:30 on Mondays. Are they general Q&A sessions, or are they projects?
No, they're generally dead about the lecture. You have questions, so that's why you should watch the movies that I upload before 10:30, so you have your questions ready by then. Nature works and what is relevant. So it's about the lecture only. Okay. And yeah, if you bring in any wishes, if you have any wishes for change or so, like we already got some, bring them in and we'll see whether we can respect them. Okay, if there are no further topics, then I would say goodbye to everyone. Thanks for tuning in, and I see you next week. Okay, bye.
Bye. Thank you.
Thanks, bye.
Or you're only concerned about